First of all, an apology, I won't be answering that question at all. I'm going to be talking about point of principle, how we're trying to address that question more than anything, I'm afraid, so apologies from the start. And I want to talk to you about the development of a mouse model of aging that we're doing at the moment. We're doing longitudinal testing of mice to look at, to try and identify good ages and bad ages across this short lifespan of a mice, which is around about 2 years in the lab. Our general hypothesis is that something's changing in connectivity, strengthening or weakening of neuronal networks, either locally within the nucleus or between brain regions as we age. It's a dogma. We think it is, but we really haven't got much evidence to prove that that is actually what is going on during aging. We're going to be doing this, and I'm going to talk to you about the individual longitudinal cognitive testing we're doing. And this is done by a postdoc, Rushdie, in collaboration with actually Paul Shazot's group in Durham, who developed the test we're doing. We're combining this with retro-AV and rabies virus to try and look at anatomically the connection between brain regions, and using high-resolution single-cell tracing in whole mouse brains, using a system called the TissueCyte, and I'll take you through that as well. But the data that I'm going to talk to you, the point of principle data that I'm going to mainly concentrate on is this. I'm going to try and convince you that we can combine an optical technique, mGrasp, with an electrophysiological technique, channel-adopting optogenetics, to map the strength of connections. It's something that I didn't necessarily believe we could do, but now I'm pretty convinced we can. And so then we're now going to be applying this to the mice that we identify in this assay. So first of all, this assay, where's my mouse? I'll just jump ahead. To show you on our assay, it's a 9-arm radial maze, made slightly more difficult. This ramp is a platform it's got to walk up before the mouse can see a visual cue at the end of the arm. It's a foraging task. They've got to find food at the end of each arm. This mouse has been doing it a while, because I can tell, because it's going up and down arms. So it knows there's food there. When you first put them in that maze, of course, they don't know what the hell's going on. And so on day 1 of the trial, we do our first trial at 4 months of age. They're operating below chance. So a lot of the time, they're just going straight back into the same arm. They don't know there's food at the arm. They don't really want to explore. It's quite a difficult task, actually. By about 7 days, they're learning, they're exploring more, and they're operating above chance levels. So what I mean by that is, essentially, they're visiting 5-arm independent arms in an exploration event, whereas by sheer chance, it would be about 2 to 3. That might not sound a lot, but this is the first time they've seen this environment. It's new. We then, of course, do this over and over again at different ages. Now, we've only done 3 time points so far with our complete data set of 16 animals. Each one's different colour there. And I just want to draw your attention to this guy here. Well, actually not a guy, they're all girls. They're all girls because we're keeping them group housed throughout their lifespan. If you do that with males, they fight. So women tend not to. The female mice tend not to. This mouse is making decisions quickly. It's got quite a quick mean decision time, and that's the time between deciding to go down in arms, and it's quite accurate by the end of the trials, by the end of 16 days of training. Quite accurate means 5 error-free movements, exploring 5 independent arms. They never go down all 9. They just don't get it. If I want to be totally anthropomorphic, mice can count to 5. That's a silly thing to say, obviously. But this mouse takes a longer time to make decisions. 4 months later, this mouse got worse, this one's got a bit better. 4 months later, this mouse got a little bit worse, and this one stayed quite well. This is a good ager, this is a bad ager. And that's what we're trying to identify. We're trying to identify over the lifespan, and we don't want them to get disease. We don't want them to get cardiovascular disease. We don't want them to get neurodegeneration. We want to track them. We're probably only going to track our cohort. Our cohort in the end will be 48 mice or so. We're probably going to stop this once we've identified at around about 16 months or so. Because we really want to see what's going on in their brains. So we want to get their brains out. We want to record electrophysiologically what's going on. So what could be changing in their brains? And this, we go back to human brain studies. This is a really good one. This is the Lothian Cohort study. Started when these individuals took their 11 pluses just after the Second World War, and they've been tracked ever since throughout their lifespan. Studies like this have shown great diversity in cognitive abilities in this group. Some age well, some age badly. And that's what we wanted to see is going on in mice. And it looks similar. Some age well, some age badly. So in human studies, what's changing? I picked this one because it's from Cambridge, and also because it's very recent. This is a Journal of Neuroscience paper from May, showing the same thing that lots of studies have shown. Our brain volumes change as we age. This is white matter scores over here, getting worse between 30 and 90, our lifespan of around about 80 years. And here, looking at some cognitive abilities. Reaction time always goes down in all of us from age 20 onwards. So that's why, of course, Usain Bolt is no longer a 100-meter sprinter. Reaction time goes down. Fluid G is your ability to perform a new cognitive task. So, for example, this morning I couldn't turn the shower on. It took me a few minutes to work out how this hotel shower worked. I'm sure a few years ago I'd have solved that quicker. That's Fluid G. Language, hopefully, isn't deteriorating. Memory, I hope, won't deteriorate too much. But I do notice some. And we're all going to experience this in our lifespan. But the question is, what's going on? Obviously, there is volume change. There are changes in the brain. But that's just MRI shape. DTI tells us a bit more about white matter integrity, diffusion tensor imaging. So this is looking at anisotropy in water molecules, their direction of movement, their diffusion, that's all. But it's probably related to myelination and the ordering of the white matter tracts. And it changes with age. So it gets better, it gets more ordered, and then it gets less ordered as we age. You can use this to predict the age of an individual. So if you take a 50-year-old man or woman, DTI will predict an age of anything from 20 to 55. You could ask them. But, you know, it shows things are changing. The question is what? Bold also shows something changing. But again, the question is what? Bold is interesting. It's an exquisite control of a hemodynamic response. So how much blood goes to different bits of your brain. And it clearly changes with age. And this is looking at, I think, resting state bold and looking at what they call functional connectivity between different brain regions. And some connectivity is maintained with age and some's lost. This has led to the idea of differentiation of the brain, either de-differentiation or differentiation. Some areas get stronger connected, some get weaker. Oddly, this is seen a lot. With aging, you see loss, you see a more bilateral response in the bold signal. It's interesting, it's really fascinating data, but for me, as a synaptic physiologist, as an ion channel person, I want to know what's going on. And that's where the mouse comes in, because I think with a mouse model, all the human data at the moment is just telling us phenomenology, telling us nothing about mechanism. We're using, first of all, we want to image the brain, and we're using this using what's called a tissue site system, which is an automated block face imaging system. The Allen Brain Atlas use a lot of this. They've got 8 of these machines. There are 2 of them in the UK, and we're fortunate that we've got one at Imperial College as part of the Centre for Neurotechnology, which is an EPSRC-funded initiative. This is some data from 2 mouse brains showing you full reconstruction of each coronal section. So what happens, of course, the tissue site, it's an automatic vibra slicer, slices the coronal section, image little bits of it at very high resolution. We can use high numerical aperture lenses, stitched together. You can see them sometimes flashing up the little squares. That's raw data. That's a 2 to 3 terabyte data file, each of those mouse brains. They're hard to handle, but they're quite dense. They're quite rich in information. The first thing we're going to be doing is taking those good and those bad learners and seeing if there's anything different about their brains, just like ours are, and see if mice over their lifespan show these sort of changes. That was actually autofluorescence. More powerfully, you can look at single cell populations, and this is a SOX14 promoter driving GFP. You can't use antibodies for this. You've got to have an intrinsic fluorescence within the brain to look at, because you've got a whole brain blocked there. But I show this because these neurons are expressed in the thalamus, is the visual thalamus, the intergeniculate, the dorsal and the ventral LGN, in the inferior and the superior colliculus there, and also in these hypothalamic regions down there. Beautiful data we can get out of this, and of course this is the other thing we can do. Because we've got micron resolution, we can single trace axons throughout the entire brain. So this is a small section of the brain. This is actually from a collaboration with Alessio De Lego in King's. He's really good at rabies virus tracing, and we're using the SOX14 as our seed and looking at the inputs onto the SOX14 neurons and tracing these cortical neurons here. And I've colour-coded two of them, this green one and this red one. This green one because we don't know what the hell it is. And it's got a classical, of course, corticothalamic projection, but it's also got this very nice layer 1-2 projection. Is that unusual for... This is the first time that anybody's tracked back neurons, the cortical neurons, onto the interneurons. But this is the sort of techniques we're going to be using. But the one I want to talk to you about most today is this. And it's the mammalian GFP reconstitution across synaptic partners, MGRASP, and how we've combined that with classical optogenetics. I say classical optogenetics now, it's like it's been around forever. Okay, so this was developed by the McGee lab at Genelia Farm. We're working with Ginny Kim, who's now at South Korea, on this for the prefrontal cortex data, which I'm not going to be able to talk to today. But this is our point of principle study, and this is the auditory system. This is the projection from the auditory cortex to the inferior colliculus, and it's ipsilateral and it's contralateral projections. We picked it because we know they're different. We know the ipsilateral has a stronger projection than the contralateral, and we wanted to see if these assays would pick that up and how sensitive they would be to that, and I was surprised. Mainly because MGRASP is amazing. Why the hell does it work? You put a presynaptic... Well, actually, I'll talk about this simple thing first. Optogenetics. We put an AAV channel rhodopsin into the auditory cortex. It projects more strongly to the ipsies and the contra. We then go and make recordings from the ipsi and the contra, and we see larger inputs in the ipsies and the contra. And these are glutamatergic inputs. We're stimulating all the inputs onto that neuron within a acute slice preparation. So if I go back, this is the blue light flash, which we picked to cover the entire neuron. So we're stimulating all the release sites onto that neuron that are coming from the auditory cortex. Only those from the auditory cortex, of course, because those are the only ones that are expressing channel rhodopsin. And we see that the responses are bigger in the ipsi and contra, and that's what we expected. Now, with these type of experiments, you get these highly polysynaptic responses, which you want to block. So you go into TTX. You also add 4AP in there to depolarize the presynaptic terminals a little bit, I assume, but I'm not really sure why. We've tried it with and without, and it seems to help if you have 4AP there. And then you get nice monosynaptic inputs. And the monosynaptic inputs also show larger responses in the ipsi and the contra, and that's the quantification there. Then we did MGRASP, and this is the spooky stuff at first. You put one fragment in the auditory cortex and the other fragment in the inferior colliculus using AAV, and only when they come together will that beta barrel incorporate itself into the other GFP fragment, and you'll get a fluorescent signal. It works, surprisingly, and it works a lot better than I expected. First of all, we see, in terms of just synapse density, higher density in the ipsi and the contra. But this is all that we expected to happen. This was good news. But there were other things that happened which we found a bit more unexpected. The one was we decided, okay, that's just GFP signal, these signals we were measuring there. What we really want to do is reconstruct single inferior colliculus neurons and count the number of synapses each neuron has, and also have a look at maybe the properties of those synapses, how they're distributed across the IC neuron. So we did that, we reconstructed and we looked at the synapse density. We saw, first of all, this, that in single neurons now, each dot is a single neuron, and we measure its surface area, the total surface area, because we got the full reconstruction, and then we count the number of synapses on it, and we can see synapse density is higher onto individual neurons in the ipsi and the contra. That's great, but we also saw this. This was looking at the surface area of the puncta. We then looked at their distribution, and I convinced myself over a few months that there was actually 3 peaks to that distribution. So we had to do quite a lot of thinking about how we bin the data and how we fit the data, and we decided there were 3 peaks. And we decided that the biggest component of the peak in the ipsi were these large synapses, whereas in the contra, the small synapses dominated. And when you looked across single neurons, you saw in the ipsi that these large synapses tended to be at the soma much more than in the contralateral projections. So you can really, the reason why we're excited about this is because this could be a lot quicker than EM, basically, to measure the number of synapses that are on the neurons. But this reminded me of mini distributions. So we looked at the mini distributions because what we noticed was an artifact of optogenetics, which we quite like. It raises presynaptic release probability. It's a whopping great unphysiological stimulus, a massive depolarization of the terminals, but it seems to cause the mini frequency to increase for some period of time. And we can measure the amplitude distribution of these minis, and we also see a similar three-peak distribution in the mini distributions. Nothing in the average peak amplitude between the two of the minis. I showed you the evoked channel adoptions, which were different, but the underlying quantal events, on average, were not statistically significant in terms of their average amplitude, but the distribution of the small, medium, and large events was apparently different. In a similar way to the mGrasp data. A larger proportion of large synapses. What we also saw was these peaks seemed to be closer together and these were farther apart. That seems to happen there as well. They're closer together and they're farther apart. It was all spookily similar. Now, we also did this. We looked to see, so this is, actually, I didn't show you the channel adoption evoked minis. So this is stimulating at 1 Hertz, 1 every second. In between these evoked events, which are in presence of TTX, of course, we see minis, and it clearly increased in frequency from the control data. This is at 0.2 Hertz, this is at 1 Hertz, and in terms of mean frequency, they increase, and then when you stop, they come back down again. Because I think you're raising the release probability in the terminal because of residual calcium. And we see no change in the decay of the minis, or their rise time, or their peak amplitude distributions, actually. It's just the frequency increases. So we're causing the release probability to increase. And we see a stronger relationship in the IPSI and the contra because there were more synapses in the IPSIs and the contra. So we think that channel adoption and stimulation is quite good for that. We had to do a few more controls. One of them was to check for multivesicular release and how that might have been affecting the distribution of amplitudes. We don't think it was affecting them at all because actually the frequencies are very low, so superimposition was very rare. And also if you raise calcium to mimic what we think is happening when the channel adopts in, you see a nice increase in frequency, no change in the rise time and the decay, and also no change in the average peak amplitude of the events. And then this was a reviewer's comment in gamma DGG, which is a low affinity glutamate antagonist, which would block spillover or events. Did absolutely nothing, as we expected, but it was useful to see. But the bottom line is a very spooky correspondence between the mini peak amplitudes and the mGrasp surface areas, which gives us faith in combining these two together to measure the strength of synaptic connectivity in defined projections. This we've finally got published in Cell, but we've got the letter just the other week. This is a collaboration between myself and Paul Chatterton at Imperial with Ginny Kim, who developed this technique at Genelia Farm. So I just wanted to mention them. And the work was done by Junho Song, who is now working with Ginny, who did the mGrasp labeling, and Diana is a PhD student of mine who did the electrophysiology of the channel adoption experiments. So now we feel we're in a position to go back to our longitudinal data. What are we going to do with the animals that we've identified as good learners or bad learners? We're going to first of all look at their brain morphology to see if there are certain brain regions that would change more than others. We're expecting PFC effect, but we don't know if there will be. And then we're going to do this mGrasp between PFC to PFC connections. And that's what's changing in those human bold studies. PFC to PFC connections are probably, that's what you might guess could be changing in those bold studies, which show this de-differentiation across the two halves of the PFC. Once again, mentioned collaborators. Bill Wisden I've been working with a lot at Imperial, who does the retro-OV. Alessio does the rabies virus tracing. This high-resolution tracing system was funded by the EPSRC, and I'm doing that in collaboration with Simon Schultz and a PhD student of mine, Gerald Moore. mGrasp we're going to carry on doing this time in PFC, And really, the overarching theme of this is hopefully in future we can say whether or not we can now map distant connections quite well. We're very good at mapping local connections in our acute slices. We do a lot of paired recordings between neurone populations. That's relatively straightforward. But we now think we're at a point where we can measure the strength of functional connectivity, electrophysiological ion channel strength between brain regions. That's it, other than to say thank you, and to remind myself that synapses are much more complicated than that. This is a beautiful watercolour by David Goodsall of the neuromuscular junction. We're getting to grips with some of the changes, I hope, that could be changing during the aging process for synaptic transmission. And that's it.
Thank you, Stephen. Questions for Stephen? Yes.
Thank you. It's a good talk. I was impressed by your early slide. You showed a good aging and a bad one. And I also noticed the good one was smart at the beginning. The bad aging is pretty lousy, at the beginning. So my question is, have you tracked the good aging and bad aging with the beginning status? What I try to get is, always the smart guy age better, stupid one like myself, have no future.
Yeah. I have exactly the same worry. So the motivation for that Lothian study, one of the things I didn't mention is one of the only predictors they had for bad performance, bad aging, wasn't lifestyle, wasn't whether or not you drank or you smoked or whatever. It was how well you did in your 11 plus. Now, I don't know about you, I failed my 11 plus.
I didn't.
I failed and I went to a secondary modern school and I really loved it. But the idea is, this is given rise to the water tank hypothesis, that what you're born with is what you die with. Because remember, our brains, neurons are very unique in that they're fully differentiated cell types, that you can't regrow neurons. I think now the idea of adult neurogenesis has been completely put to bed. You cannot regrow. So, in answer to your question, we can't track before 4 months. We can only start at 4 months. What we're seeing is rank orders are preserved. So those guys that were smart at the start are smart, that were quick at the start are quick at the end, and those that are smart at the start, that were slow remain slow. Whether or not we see the bad aging consistently in the slow processes, we don't know yet. But I thought if I guess it might.
Matt.
So you're using genetically identical mice.
Yes.
So what are your ideas about what might account for the variance that you see?
Absolutely.
These are all kept in the same cages as well.
Absolutely, utterly no idea. Yeah. Don't know. I was first of all just fascinated to see if there would be variability in what is an inbred strain. They're an inbred. They're all females. They're all inbred. They're all litter mates. They're actually all from the same litter. But we still see clear differences which are maintained throughout their lifespan. What we don't know yet is what predicts good or bad
Do they
aging.
have social structures?
Not sure. I'm sure they do. I'm sure this is what Rushdie thinks that, actually he thinks the more submissive ones might be better ages than the more dominant, but we're not sure. Yeah.
Yes.
Can you learn new tasks? Because now we tested on the same task over. Are they good or bad at learning new things?
The only other thing we're doing is novel object recognition. That's the only other test. Rushdie is a hero. I did not realise when I started this how difficult it would be. He can't be with us. He could never come to meetings at this time of year. He's now got 2 months of solid behavioural testing on huge numbers of animals just to do that. It's silly, the amount of time that you have to dedicate to the longitudinal testing. So we can't really do much more. And I quite like the test anyway. It's difficult, they can't do it, so we haven't got a ceiling effect, so we can see improvement and we can see decline. So I'm quite happy with it.
So in terms of the Ipsian contralateral synapses, you concentrate on the presynaptic, in terms of release probability or changes in that. Postsynaptic synapses was the same. Do you have any spine data? These synapses form onto spines, or spines equivalent?
No, they're quite smooth. They're quite
smooth. Okay.
Yeah. These guys are quite smooth, very small. They're only about 15 picofarads. So we were lucky that you can space plant them. So you get quite good minis. I don't think this is a good system for all networks.